let's discuss a paper from the Mock Championship 2026 by Fast Papers, and this is Cambridge IGCSE Biology Paper 4, and the total mark is 80. Starting with question 1, figure 1.1 shows part of the human gas exchange system. So in this diagram we can see that there is a cartilage on top of which there is a structure labeled C, and below that, starting the branching section, is a structure labeled B, and A is labeled by smaller, more branched out structures. So identify structures A, B, and C shown in figure 1.1. So A will be bronchioles, B will be the bronchi, and C will be trachea. State the function of the cartilage shown in figure 1.1. So the function is cartilage keeps the airway open and prevents the airway from collapsing. So it basically acts as a support system so that air can pass in and out without it bending or getting collapsed in any sort of way that interrupts the gas exchange procedure. Number 3, explain how inspiration occurs. So all these points over here account for 1 mark. So all you need to do is mention any 4, but aim to mention a little bit more so that in case one is correct, the other one gets marked, but you will likely get marked on the first 4 points. So in inspiration, air is inhaled, and to facilitate this, our gas exchange system does the following. The volume in the thorax, lungs, or chest, either one from here, increases, the rib cage is lifted, and the diaphragm contracts. Then the external intercostal muscles contract and internal intercostal muscle relax. So you will understand these points a little bit more if you had a diagram of the gas exchange system in front of you, so you can look it up while watching this. And pressure in the thorax, lungs, or chest decreases. Air is drawn in to equalize the pressure between atmosphere and lungs. This happens because air pressure in lungs is lower than in the atmosphere, so air moves down a pressure gradient. Part B, cystic fibrosis is a lung condition in which the mucus in the airways is thicker and stickier than normal. So in 1, the airways are lined with 2 types of cells that defend the body against pathogens and dust particles. State the names of these 2 types of cell and describe how they protect the body. So the 2 types of cells are goblet cells and ciliated cells, but you could also mention lymphocytes and phagocytes. And depending on which 2 you have mentioned, their function is described as goblet cells secrete mucus to trap particles and pathogens. Ciliated cells move the mucus up the airway. Lymphocytes produce antibodies, and phagocytes engulf the particles or pathogens and carry out phagocytosis, which is basically taking in the pathogens or particles and taking it away from the... So what it does is, goblet cells, the mucus secreted by goblet cells, they trap the particles. Think about dust, for example, and then the ciliated cells move the mucus up the airway so that it can be moved into your stomach and removed from the airway so that it doesn't to your lungs. B2, suggests why having thick, sticky mucus in the airways may make it harder for a person to exercise. So, this these lines over here kind of indicate the mark scheme points. So again, any four of these will get you 4 marks. So thick sticky mucus acts as a blockage in the airway. This reduces the surface area available for gas exchange to occur or diffusion. Therefore, the supply of oxygen becomes less, or the supply of oxygen is slower, along with a slower removal of carbon dioxide into the blood and muscles. This results in less aerobic respiration and more anaerobic respiration to take place. Energy is released less, as well as less ATP is produced for muscle contraction. As a result, tiredness can be observed, making exercise difficult. So, in C, cystic fibrosis is an inherited condition in humans caused by a recessive allele of a gene. There are two alleles for this gene. The allele for no cystic fibrosis is represented by the letter F, and the allele for cystic fibrosis is represented by the letter F. So, CAPS is usually dominant, and the smaller case is recessive. Two heterozygous parents wanting to have a child complete the genetic diagram to predict the probability of these parents having a child with cystic fibrosis. So the parental phenotypes are no cystic fibrosis for both of them, and the genotypes are F, caps F, and smaller case F for both. So, so the dominant allele is um, is preventing the cystic fibrosis in them, but they are heterozygous, so there's 1 in 4 chances of being passed on, which we are going to look at now. So the gametes are caps F, small f, caps F, and small f. So parent 1 has these two, parent 2 has these two, they are the same. So what do we get here is one of them will have both the dominant alleles, which means absolutely no chance of cystic fibrosis, and there's 50% chance of them being heterozygous as well, and there's 25% chance of the offspring having cystic fibrosis, which is indicated by 2 lower cases of F. So these are the possible genotypes. These are all 25% chance, but since these are the same, this makes it 50%. But in 70% of the case, there will be no cystic fibrosis, and only in 1 out of 4 cases there might be. So the probability is 25, or 1 out of 4. It's important to also note that the allele is indicated by either F or F, and you should not use XX or XY here, because it confuses the whole concept of a gene being something that is not related to gender. Okay, in number 2, the temperature inside a person's body was monitored and recorded. Figure 2.1 shows the results. So there is time on the x-axis, and on the y-axis there is body temperature. So in A1, it says use the information in figure 2.1, calculate the temperature range for the internal body temperature of the person. So it says calculate the temperature range, which means you don't only state a range, you calculate something out of it. So the maximum here is at 38, okay, and the minimum here is 36. So you subtract the higher value from the lower value, of course, and then get the range, which is 2 degrees Celsius. And the mark is both for mentioning 2 and degrees Celsius. So if you mention just 2, you won't really get the mark. Okay, on figure 2.1, draw a line to show the set point for the internal body temperature of a person. So you draw a line straight from here and indicate it, which is 37 degrees Celsius. In 3, the maintenance of internal body temperature is an example of homeostatic control. State the name of the mechanism for homeostatic control. And it's called negative feedback, because you are getting a feedback that the temperature is either too high or too low, and it needs to be set to normal back. So here, if it's too high, you need to bring it back to 37. If it's too low, you need to raise it up to 37. So this is the process of negative feedback and how the control is. 4, on figure 2.1, region F shows a change in body temperature. Explain how the body causes the change in body temperature shown. So I explained a little bit of that in explaining negative feedback. So we get into detail on this question. So region F is showing a change in temperature from low to high. So this is the F. So it's moving from 36 to 37 and then 37 to 38. So what's happening here is the brain detects this low temperature and signals the constriction of arterioles, or this process is also known as vasoconstriction. When this happens, blood flow to the skin surface or capillaries reduces, causing increased skeletal muscle contraction, and shivering occurs. So when it falls below 37, this is what happens. And then the rate of respiration increases and hair erector muscles contract, which means hair stands upright to trap a layer of air. And this acts as insulation, working to increase the body's temperature. So it's going from 36, 38, and then it's falling back to 37. So the body responds to this low temperature by raising it, and then it slowly comes down to the normal temperature. Okay, part B, figure 2.2 shows a cross section of human skin. State the letter of the structure shown in figure 2.2 that produces sweat. So out of all this, this is the obvious one. This is the sweat gland. And then state the name of the structures B and C shown in the diagram. So B here is a neuron. In C we have the hair erector muscle, which also responds to the change in temperature by controlling how the hair would respond. Okay, in figure 3.1, it shows a structure of the pathogen that causes cholera, and state the name of the kingdom that includes the pathogen shown in figure 3.1. So the answer will be prokaryote. And then in table 3.1 shows some of the names, functions, or uses, and identifying letters of the parts labeled in figure 3.1. So in this table, few of the things are already given, which is flagellum, movement of the pathogen within a medium, and It's indicated by the letter H. Then we have C given, and C is the cell membrane. I labeled it wrong, my bad. So what the cell membrane does is it controls what goes in and out of the cell. Okay, and it's important to understand that the first border outside the cytoplasm, or this part of the cell, is the cell membrane, and right outside the cell membrane will be the cell wall. And you might want to look it up what this structure is in case you're confused, which is labeled by E here, but this is not the cell wall. This facilitates movement or controls the transfer of genetic material. So then we have protein synthesis given, and which is done by ribosome clearly. And since it's a small structure, it's obviously indicated by F. And we put F over here. And then we have the cell wall, which is A, like I said. So what it does is it prevents the cell from bursting, and it acts as a support. And lastly, we have used by humans in genetic modification. So in biotech, this is often needed for genetic modification, and it's the name of it is plasmid. It's this area over this structure over here, and it's indicated by the letter G. Okay, and B, describe how a person becomes infected with cholera. So the only way is drinking contaminated water or consuming food that has been handled with contaminated water. And describe how this and see, this is not the question here. It says something like the people infected with cholera get very dehydrated, and explain how cholera causes dehydration. So let's break this down. When infected food or water is ingested, cholera producing bacterium produces a toxin which causes secretion of chloride ions into the small intestine. The loss of ions increase water potential within the cell, making water move out of the cell and down a water potential gradient via osmosis. So as a result, water is lost from the body through watery feces or diarrhea. For this question, vaccination can help to prevent the spread of diseases such as cholera. Outline the process of vaccination and explain how it can prevent the spread of diseases. So there are a lot of points from the mark scheme. You have to mention any 5, just which you can mention, but any point outside the mark scheme is usually not accepted, even if you are correct. So I mentioned all of the points, and I'm going to explain it right now. A vaccine contains weakened or dead bacteria or pathogen which carry antigens. So antigens stimulate an immune response in the body, causing lymphocytes to make antibodies. Then these antibodies bind to the antigens. This happens because the antibodies that are made have a shape complementary to the antigens. So the lymphocytes make this antigen specific antibody so that those bacteria or pathogen can be destroyed. This causes our body to retain this immune response by forming memory cells. So Once this happens, your lymphocytes produce this antigen specific antibody. Your body remembers this process, it remembers making these antibodies, and you form memory cells so that the next time anything near the dead bacteria or pathogen comes in, you will have an instant immune response. Therefore, long long-term immunity is achieved. Since cholera is transmitted from person to person, mass vaccination programs can prevent spreading of such diseases. Vaccination part is really important um, because even if you get vaccinated, you're still carrying the pathogen and you can pass it on to others who might not be vaccinated. So this won't be as effective as preventing the spread. So that's why mass vaccinations are always important. Okay, so let's move to question 4. In figure 4.1 is a diagram of the nitrogen cycle. So in A1 it says state the names of processes A and B in figure 4.1. So A will be denitrification, because this is a process where nitrates and nitrogen gas escapes from the soil and goes back into the atmosphere. And B will be nitrification, because this is a process in which energy from ammonia is taken in by bacteria to produce nitrates. So it's shown by nitrate ions in soil, and this is obviously the ions and gas going back to the atmosphere. Okay, state 2 ways that nitrogen fixation can occur. So it's lightning and bacteria. And say the name of compound Y in figure 4.1, and it will be ammonia or ammonium ions, because that is what is being broken down into nitrate ions. Okay, so last one is state the name of an organ in animals where assimilation and deamination of amino acids occur, and it is the liver, because in liver is where assimilation of digested materials are taken place. Okay, so we have another one from A. Describe the process of deamination. Deamination is the removal of nitrogen containing parts of proteins or amino acids and forming urea. Very simple. It's basically 1 mark for the removal and 1 mark for forming urea. Okay, so this will be B. Complete the sentences about pollution of the air by writing the appropriate words or phrases in the spaces provided. Okay, so methane is a pollutant gas that contributes to the enhanced greenhouse effect and climate change. Methane is released into the atmosphere from cattle and from the farming of rice crops. This is because herbivores feed on rice crop, because this is where chemical energy from this crops is directly taken in by the primary consumers. Now the second part, dash is a gas that is absorbed by trees in the process of dash. So it's basically carbon dioxide, because trees are always taking in one gas, and they do this to photosynthesize. This gas is released into the atmosphere by the burning or combustion of fossil fuels, where it also contributes to climate change. Besides other living beings respiring, dash of fossil fuels will obviously be burning or combustion. In C, explain the process of eutrophication in aquatic ecosystems. So again, I wrote everything in detail and included every single point from the mark scheme. So let me break it down. Eutrophication happens when untreated sewage um, or fertilizers enter the aquatic ecosystems. This increases the availability of nitrates or ions or nutrients, you can mention any one, causing increased algal bloom. Algae reduces the amount of sunlight that enters the water bodies. So it acts as a barrier on top of the water bodies and makes it harder for sunlight to penetrate through. This makes photosynthesis difficult for rooted aquatic plants, like the ones under the water bodies, causing them to die. And dead plants decompose and bacteria feed on them, increasing bacterial population. And as they increase in number, they also respire aerobically and they use up more oxygen from the water, and therefore the lack of oxygen present in the water reduces, and fishes and other organisms that take in the oxygen also start to suffocate and therefore die or have to migrate to another water body if they are able to. Okay, let's move on. Number 5. Some algae are single celled organisms that can photosynthesize. Their cells contain the pigment chlorophyll. State the name of one mineral ion that is needed to make chlorophyll. And this is magnesium ions, or you can just write magnesium. B. A student investigated the effect of light on photosynthesis in algae using hydrogen carbonate indicator solution. Carbon dioxide is an acidic gas. Table 5.1 shows the color of hydrogen carbonate indicator solution at different pH values. So there's a pH of 7.6, 8.4 and 9.2. In 7.6 it's yellow, in 8.4 it's red, in 9.2 it's purple. So B1, 2 test tubes were set up at the same time. Both contained algae and hydrogen carbonate indicator. At the start of the investigation, the hydrogen carbonate indicator was red in both test tubes. So this is a control, and one test tube was placed in a dark and one test tube was placed in the light. Explain these results. Okay, so algae photosynthesize when light is available. Okay, so they cannot do so in the dark. During photosynthesis, carbon dioxide is absorbed. So obviously plants take in carbon dioxide to photosynthesize. So when carbon dioxide increases due to photosynthesis, the mixture in the test tube becomes alkaline. So carbon dioxide is an acidic gas, right? So it's taken in and it increases the pH. So this is indicated by the pH of 9.2 and the color purple. So when carbon dioxide is used up, it becomes more alkaline than acidic and the color is purple, so and the pH is 9.2. Algae also respire in the dark where carbon dioxide is released. So in the dark they have to take in oxygen and release carbon dioxide. So naturally what happens is when there is carbon dioxide released, it will become acidic, so the pH will go down. This respiration reduces the pH and the mixture becomes acidic. This is indicated by the pH of 7.6 and the color yellow. Okay, in number 2, figure 5.1 is a graph showing the effect of light intensity on the rate of photosynthesis. State which factors could be limiting the rate of photosynthesis at X and at Y in figure 5.1. So at X, the limiting factor is light intensity, and at Y, there are multiple limiting factors. So you mention any two of these, and mentioning one won't really get you 3 marks, because 1 mark is for this and 2 marks are for either two of these. Okay, so C says starch and sucrose are made by plants after photosynthesis. State the name of the tissue that transports sucrose around the plant, and the name of one other biological molecule that is transported in this structure. So obviously it's phloem, and this also transports amino acids. So this is the last question of this paper. Describe how starch is broken down in the human digestive system so that it can be absorbed into the blood. So starch is broken down through mechanical digestion, which occurs in the mouth using teeth. Then churning of the chewed food occurs in the stomach. In the mouth, salivary glands secrete amylase, which breaks starch down into maltose. So chewing also increases the surface area for efficient enzyme activity. And then in the stomach, chemical digestion takes place, which breaks down insoluble molecules into smaller, more soluble molecules. And after passing the stomach, maltase found on the epithelium lining of the small intestine breaks down maltose down to glucose. The optimum pH for this enzyme is neutral. So when it's too acidic or too alkaline, it will get denatured. So what happens is starch is broken down into amylose, and then amylose is broken down into maltose, and then maltose turns into glucose. Okay, thank you for watching. Best of luck for your exam.